Hi, my name is Cheryl O'Donnell. I'm an identifier with USDA APHIS PPQ in San Diego, California. My discipline is entomology, and my specialty is Thysanoptera systematics and taxonomy. I'm standing in the National Museum of Natural History's Thysanoptera collection, hosted by USDA's Agricultural Research Center in Beltsville, Maryland. This is one of the most extensive Thysanoptera collections in the world, dating back to the early 1900s. Each of these boxes contains 100 microscope slides of specimens of thrips from all over the world. There are over 110,000 slides in this collection, representing 3,300 records. Today I'm going to demonstrate how to prepare and slide mount your thrips specimens. I will include larvae and all adults of both suborders, Tubulifera and Terebranchia. I hope these demonstrations are helpful to you in the future for your identification purposes. I just finished field collecting. I'm now sitting at the dissecting scope, where I've gathered all of the tools and chemicals to create slides of the specimens I just collected. So let me introduce you to all of those tools and chemicals that we'll be using. First, we have a watch glass for sorting our field collection. We have a series of micro tools, 2 different types of forceps, several pipettes. We have a glass rod for dropping our media onto our slides and our cover slips, a series of watch glasses for the chemical preparations, chem wipes to keep our tools and our watch glasses clean. We have our slides, our cover slips, our centering stage, 2 types of pens to write on the glass watch glasses or on the slides. We have a series of chemicals, potassium hydroxide, ethanol, balsam, Hoyer's, and clove oil. We have our hot plate to cure our slides, a tray to put our slides in once they're cured, and we have our mailing envelopes for our slides. We will be using most of these in our slide preparation. Tubulifera are generally large, dark-bodied thrips, requiring additional time and care during the maceration process prior to slide mounting. The time required for maceration is dependent upon the size and color of the specimen. Large, dark-bodied thrips may require 24 hours in 5% KOH solution, whereas a small, light-bodied individual may only require a few minutes. The latter would also include larvae at a 5 to 10% potassium hydroxide solution. I placed several Tubulifera individuals into 5% solution of KOH, and I left them on a warming plate at 35 to 40 degrees Celsius for 24 hours. Today I will process them onto the microscope slides. Before I remove the individual from KOH, I ensure that the body contents are clear and the body is translucent and that the wings are spread. If the body contents are not clear, then I would use my micro spatula to gently pump the body to flush out the body contents. This pumping will help to exchange potassium hydroxide with the body contents. If the wings are not spread during maceration, then I would manipulate the wings into the desired position about 10 to 15 minutes before I remove the individual from the KOH. As you can see, this individual is translucent and the wings are spread to the desired position. Therefore, we can proceed to the dehydration process. I will lift the specimen out of the KOH and place it into a small watch glass with 70% ethanol solution, leaving the specimen in the solution for about 15 minutes. Now we will transfer the specimen into 95% ethanol and again massage the specimen as we did in the first step. Leave the specimen in 95% ethanol for 10 minutes. The specimen has been adequately dehydrated in ethanol and is ready for transfer into the clove oil for the last dehydration step. Clove oil will remove the ethanol and any remaining water from the specimen. Following the last few steps, we will again massage your specimen to pump ethanol out of the body and pump clove oil into the body. Leave the specimen in the clove oil for a few minutes to 30 minutes. The specimen has been in clove oil for an appropriate amount of time, and the dehydration process is complete. I will now prepare the slide and cover slip for slide mounting the specimen. The first demonstration, I will show you my preferred slide mounting method, which is to place the specimen ventral side up onto the cover slip in Canada Balsam, and by placing the slide on top of the specimen, media, and cover slip. Notice that I have placed the cover slip onto a centering stage in order to center the cover slip, media, and specimen. Using the glass rod, place a small drop of Canada Balsam onto the cover slip and spread it out. With the micro spatula, lift the specimen out of the clove oil and place it into the Canada Balsam. Press the specimen down into the media so that it is against the cover slip. Ensure that the wings are spread out to the sides. Once you are satisfied with the position of the specimen, then slowly place your slide onto the cover slip by lowering it gently from one side. Once the slide is on the cover slip, immediately turn your slide with the attached cover slip over. At this time, you can manipulate the specimen, if necessary, by gently pressing down on the cover slip. Place your prepared slide onto the hot plate with the temperature set at 35 to 40 degrees Celsius. The second method that I will show you for slide mounting today is the method that is referred to in most protocols for slide mounting soft-bodied insects, which is mounting the thrips onto the slide and placing a cover slip on top of the specimen. Using a glass rod, place a small drop of balsam onto the slide and spread the balsam out. Be careful not to spread the balsam farther than the cover slip will cover. Using a spatula, remove the specimen from the clove oil and place the specimen into the balsam on the slide, dorsal side up. Orient your slide so the specimen is positioned with its head towards the bottom of the slide and you. Arrange the specimen with wings spread out to the sides. Once the specimen is in position, place the cover slip on top of the balsam with the specimen in the center. Occasionally, you may need to gently tap the cover slip to start the balsam movement to adequately fill the entire cover slip. The slide is now ready to be placed onto the warming plate or oven set at 37 degrees Celsius. Many species of the Terebrancha are light and small in size. The amount of time you leave the specimen in the chemical maceration and dehydration process will depend on the size and color of your specimen. The specimen in this demonstration is very small and light in color, so I left the specimen in 5% potassium hydroxide solution for about 20 minutes. The specimens have cleared nicely and the legs and wings are in the desired position for slide preparation. Using the micropipette, remove the specimen out of the KOH and place it into the watch glass containing 70% ethanol. Leave the specimen in 70% ethanol solution for about 15 minutes. Using the micropipette, transfer the specimen into 95% ethanol. This process will take approximately 10 minutes. The specimen is now ready to be transferred to clove oil for the last dehydration step. I'll leave the specimen in clove oil for approximately 10 minutes. Now we are ready to move the specimen from clove oil into the Hoyers for a temporary slide mount. Place the cover slip onto the centering stage and place a small drop of Hoyers onto the cover slip. Place the specimen onto the cover slip ventral side up, making sure the wings are spread out to the sides. Push the specimen down into the Hoyers and ensure that it is in the proper position. Carefully place the slide onto the cover slip, lowering the slide slowly and gently down from one side. This helps to prevent air bubbles in the Hoyers. Quickly turn the slide with the attached cover slip over so that the cover slip is on top of the slide. This is the time to gently manipulate the cover slip to move the specimen if necessary. Place your prepared slide onto the hot plate with the temperature set at 35 to 40 degrees Celsius. This next demonstration will explain a temporary slide mount for larvae. I place the cover slip onto the centering stage and place one drop of Hoyers onto the cover slip and spread it out. Move the larvae directly from the collecting ethanol and place it onto the Hoyers. Push the larvae down into the Hoyers and ensure that the larvae is ventral side up. Now place the slide onto the cover slip. Immediately turn the slide and cover slip over and manipulate the larvae if necessary. I prefer this direct method for quick identifications and for retaining natural color of the specimen. Thank you for joining me today on the demonstrations for preparing and slide mounting thrips. Now that you know how to use these techniques, I hope they're helpful to you in preparing your thrips for identification and archiving.